Prions are unique infectious agents composed of misfolded proteins. Unlike other pathogens, prions lack nucleic acids, meaning they don't carry genetic information in the form of DNA or RNA. The term prion is derived from proteinaceous infectious particle. The primary characteristic of prions is their ability to induce other normal proteins to also misfold. This misfolding leads to a chain reaction that disrupts cell function and ultimately causes cell death. Prions are associated with several fatal neurodegenerative diseases known as transmissible spongiform encephalopathies, TSEs, which include conditions like Creutzfeldt-Jakob disease in humans, Scrapie in sheep, and bovine spongiform encephalopathy, commonly known as mad cow disease, in cattle. Here's a detailed breakdown of prion characteristics. Structure: prions are misfolded versions of a normal protein, usually the prion protein, PRP. The normal PRP is found on the surface of cells and is thought to play a role in cell signaling and maintaining cell health. Function: the exact function of normal PRP is not fully understood, but it's believed to be involved in processes like cell adhesion and intracellular signaling. Replication: prions replicate by binding to the normal versions of the protein and causing them to change their shape to the abnormal form. This process doesn't involve the replication of genetic material but rather a conformational change in the protein structure. Diseases: prion diseases affect the brain structure, leading to symptoms such as memory loss, behavioral changes, and coordination problems. These diseases are progressive, have no known cure, and are always fatal. Prions are resistant to standard sterilization methods and can remain infectious even after procedures that would typically destroy other pathogens. This resistance is due to their proteinaceous nature, which isn't easily broken down by heat or radiation. Research into prions has expanded our understanding of proteins and diseases, and it continues to be an active area of scientific inquiry. Prion diseases, also known as transmissible spongiform encephalopathies, TSEs, are a group of progressive neurodegenerative disorders that affect both humans and animals. Here are some of the prion diseases identified to date: human prion diseases, Creutzfeldt-Jakob disease, CJD, a neurodegenerative disorder with characteristic clinical and diagnostic features. Variant Creutzfeldt-Jakob disease, VCJD, related to mad cow disease and affects younger people, possibly contracted by eating infected meat. Gusman-Strauss-Leskinka syndrome, a genetic disorder that leads to progressive neurodegeneration. Fatal familial insomnia, a rare genetic disorder that affects the brain's ability to sleep, leading to severe physical and mental deterioration. Kuru, found in New Guinea, caused by consuming human brain tissue contaminated with infectious prions. Animal prion diseases, bovine spongiform encephalopathy, BSE, also known as mad cow disease, it affects cattle. Chronic wasting disease, CWD, affects deer, elk, and moose in some areas of North America, South Korea, and Norway. Scrapie, affects sheep and goats. Transmissible mink encephalopathy, affects farmed mink. Feline spongiform encephalopathy, affects cats. Ungulate spongiform encephalopathy, affects other hoofed animals. These diseases are characterized by long incubation periods, spongiform changes in the brain, and a lack of inflammatory response. They are usually rapidly progressive and always fatal. Prions cause disease by inducing a structural change in the normal, healthy proteins found in the brain. Here's a step-by-step explanation of the process. Normal prion proteins: the cell surface has normal prion proteins, PRPC, that are involved in cellular functions. Conformational change: when a misfolded prion protein, PRPSC, comes into contact with a normal PRPC, it induces the normal protein to also misfold into the PRPSC form. Protein accumulation: these abnormal proteins accumulate in the brain, forming aggregates or plaques that the body cannot easily clear. Cell damage: the accumulation of misfolded proteins disrupts normal cellular processes, damages nerve cells, and leads to the characteristic spongiform changes in the brain tissue. Neurodegeneration: as more cells are damaged and die, neurological symptoms develop, such as memory impairment, personality changes, and difficulties with movement. This process is unique because it doesn't involve the replication of genetic material, which is a common feature of other infectious diseases. Instead, prions propagate by changing the conformation of neighboring proteins. 12. The exact mechanism of how the misfolded proteins cause cell death is still not fully understood, but the damage they cause is extensive and leads to the severe symptoms and eventual fatality associated with prion diseases. The difference between PRPC, normal cellular prion protein, and PRPSC, Scrapie protein, lies in their structure and function. Structure: PRPC is primarily composed of helices, while PRPSC has a higher content of sheets. 1. This structural difference is crucial as it affects the protein's properties and its ability to induce disease. Solubility and digestibility. PRPC is soluble in water and sensitive to protease digestion, meaning it can be broken down by enzymes. In contrast, PRPSC is insoluble and resistant to protease, making it difficult to break down too. Function. PRPC is involved in normal cellular processes, although its exact functions are not fully understood. PRPSC, however, is associated with disease. Its accumulation in the brain leads to neurodegenerative disorders. 3. Infectivity. PRPC is not infectious, but PRPSC can propagate its misfolded form to other normal prion proteins, leading to a cascade of misfolding and disease. 4. The transformation of PRPC into PRPSC involves a conformational change that increases the sheet content, which is thought to underlie the infectious nature of prions. The incubation period for prion diseases can vary widely depending on the specific disease and the individual. It is the time from when a person or animal is first exposed to the prion until symptoms appear. Here are some general guidelines. Kuru. The incubation period can be very long, with cases reported ranging from 34 to 41 years as a minimum, and a likely incubation period of 39 to 55 years. 1. Creutzfeldt-Jakob Disease. Creutzfeldt-Jakob Disease (CJD). The incubation period can range from months to several years. The disease is usually fatal within 6 months to 5 years after the onset of symptoms. 23. Variant Creutzfeldt-Jakob Disease. Variant Creutzfeldt-Jakob Disease (VCJD). This variant, related to BSE or mad cow disease, has a longer incubation period than classical CJD, but exact time frames are difficult to determine. It's important to note that these periods can be influenced by genetic factors, such as the polymorphic codon 129 of the PRP gene, which can affect susceptibility and incubation time. 1. Prion diseases are distinguished by their long incubation periods, which make them particularly challenging to study and manage. The diagnosis of prion diseases is complex and typically involves multiple steps, including clinical evaluation, imaging, and laboratory tests. Here's an overview of the diagnostic process. Clinical evaluation. Doctors will assess symptoms and medical history, looking for signs of rapidly progressive dementia and neurological decline. Imaging tests. MRI scans can reveal patterns of brain atrophy that are suggestive of prion diseases. Electroencephalogram. EEG and EEG may show characteristic changes in brain activity associated with prion diseases. Laboratory tests. Blood tests can help rule out other conditions, and more specialized tests can detect markers associated with prion diseases. Biopsy or autopsy. The definitive diagnosis of a prion disease is usually made by examining brain tissue, either from a biopsy in living patients or post-mortem during an autopsy. 1234. It's important to note that currently, there is no single diagnostic test for prion diseases in living humans or animals. Researchers are working on developing new techniques to increase the sensitivity and specificity of prion protein detection in body fluids and to identify novel surrogate markers. Prions, the infectious proteins responsible for a variety of neurodegenerative diseases, were discovered through a series of scientific observations and experiments that spanned several decades. The story of prions begins with the study of Scrapie, a disease affecting sheep, which was known about for over 250 years. 1. However, the modern understanding of prions and their unique characteristics as infectious agents without genetic material began with the work of Stanley B. Prusiner. In the 1960s and 1970s, Prusiner, a neurologist and biochemist, became intrigued by rare neurodegenerative disorders known as spongiform encephalopathies, which included Creutzfeldt-Jakob disease in humans and Scrapie in sheep. These diseases were characterized by progressive dementia and a spongy degeneration of the brain too. Prusiner's key discovery came in 1982 when he isolated an infectious agent from affected brains and found it to be composed solely of protein. He named this agent a prion, derived from proteinaceous infectious particle. This was a revolutionary concept because it challenged the prevailing dogma that infectious agents needed nucleic acids, DNA or RNA, to replicate. 3. The idea that a protein alone could be infectious was met with skepticism, but Prusiner's continued research, which included identifying the gene encoding the prion protein and showing that it was also present in healthy individuals, gradually gained acceptance. By the mid-1990s, the prion hypothesis was widely recognized, and in 1997, Prusiner was awarded the Nobel Prize for Physiology or Medicine for his discovery of prions too. The discovery of prions also had a significant impact on public health, particularly with the emergence of mad cow disease, bovine spongiform encephalopathy in the UK in the 1980s and its potential link to a new variant of Creutzfeldt-Jakob disease in humans. Prusiner's work provided the foundation for understanding these diseases and addressing the public health concerns they raise too. Prion research continues to be important today, not only for understanding and treating prion diseases but also for insights into other neurodegenerative diseases, such as Alzheimer's and Parkinson's, which share some characteristics with prion diseases. That was all. Goodbye.